Hello, this is Gina Piscatelli with the second respiratory system lecture for Anatomy and Physiology 2 during week 7. In this presentation, I'll discuss how the blood transports gas, and of course the gases that we're interested in are oxygen and carbon dioxide. It turns out that gas transport in the blood isn't that straightforward because neither oxygen or carbon dioxide is very soluble in water, therefore not soluble in plasma, which is largely water, or cytoplasm of the red blood cells. So there has to be some mechanisms available in the blood to transport these gases. And there's two that are the most important. One is a protein called hemoglobin, present in red blood cells, and the other mechanism is an enzyme called carbonic anhydrase, also present in red blood cells. And what that enzyme does is it converts carbon dioxide into a more soluble form, carbonic acid, which dissociates it's a polar molecule, so it's soluble in water, but it dissociates spontaneously into two ions. So hemoglobin is what will take care of oxygen for us. Hemoglobin can also bind to carbon dioxide, though, although not very much of it, not very much of the carbon dioxide we produce is actually transported by hemoglobin. In addition to just being able to carry these gases because of these mechanisms, the mechanisms help with loading and unloading of the appropriate gas at the lung as well as at the systemic tissues. Because of how these two mechanisms work, they're ideal for dropping off oxygen at the systemic tissues, picking up carbon dioxide, and then at the lung, picking up oxygen and dropping off carbon dioxide. All right, let's start with an overall view of how much oxygen is transported by which mechanism and how much carbon dioxide is transported by the various mechanisms. Oxygen is transported in two ways by the blood. It turns out that some of it is just dissolved freely, even though it's not very soluble, of course, in the plasma. But that's only 1.5% of all the oxygen that's in your blood. The majority of it, in fact 98.5% of the oxygen in your blood, is carried on hemoglobin. It is bound to hemoglobin. So oxygen is transported in two ways. Carbon dioxide is transported in three different ways. Some of it is dissolved in the plasma, even though it's not very soluble. That's only 10%. 20% of it makes its way into the red blood cell and binds to hemoglobin. But the majority of it travels in the plasma in an altered form, a more soluble form, and that's bicarbonate ions. We're going to start with oxygen transport and how hemoglobin works to deliver oxygen to the tissues and drop it off and pick it up in the lungs. So let's look at some structure of hemoglobin to begin with. When hemoglobin is not bound to either gas, it is called deoxyhemoglobin. And I guess you could call its molecular formula HHB. H for a hydrogen ion, because that's what it's bound to, and HB for the hemoglobin. So deoxyhemoglobin is the form of hemoglobin not bound to either gas. Instead, it's bound to a hydrogen ion. Oxyhemoglobin on the other hand, is hemoglobin that is bound to oxygen. And when deoxyhemoglobin and oxygen gas react together, we end up with oxyhemoglobin, but a displaced hydrogen ion. And that's going to play an important role later. This hydrogen that's been displaced off of deoxyhemoglobin. Now there's two other forms of hemoglobin, both bound to some sort of organic compound, carbon dioxide and carbon monoxide. So carbaminohemoglobin is HbCO2. That means the hemoglobin is bound to carbon dioxide. And the name might not make much sense to you, but carb is for carbon dioxide, of course. The amino part has to do with the fact that the carbon dioxide binds to the polypeptides of the hemoglobin. And remember, there are 4 chains, and we'll look at that again. But carbaminohemoglobin is hemoglobin bound to CO2. Carboxyhemoglobin is bound to carbon monoxide. You've heard of carbon monoxide, I bet, as a poisonous, odorless gas. And we'll see why that's important in our discussion in a little bit. I know it's not that important for our metabolism. In fact, it is a poison, but it's interesting. Okay, let's review hemoglobin. You've seen this molecule before when we talked about sickle cell anemia and red blood cells in general. It's composed of 4 polypeptide chains. We could call them globin chains. And each one of these can bind to 1 carbon dioxide. So that means that 4 carbon dioxides could bind to 1 hemoglobin molecule. There's also 4 heme groups embedded in each one of these polypeptide chains. And these heme groups contain iron in an oxidized form. And each of the irons or heme groups can bind to 1 oxygen. So again, we can get a total of 4 oxygens for each hemoglobin. What's interesting about this discussion, I guess, is that because oxygen binds to the heme, whereas carbon dioxide binds to the polypeptide chain, hemoglobin can carry both oxygen and carbon dioxide simultaneously. And I don't know what you call that molecule. We're going to discuss hemoglobin in terms of saturation, how much hemoglobin in your body is saturated with oxygen. And when we do that, think of just 1 hemoglobin molecule as representing all of the hemoglobin molecules in your blood. So, fully saturated hemoglobin, I guess, is reported mathematically as 100% saturated hemoglobin. It means that 100% of the hemoglobin molecules in your blood, or in your red blood cells in your blood, carry oxygen on all 4 heme groups. But there's some hemoglobin that's probably partially saturated, usually, and depending on where you are in the bloodstream. And partially saturated hemoglobin means that there's only 1, 2, or 3 oxygens on a hemoglobin molecule on average. So we usually report this more in percentages. So, for example, 75% hemoglobin saturation means that on average, every hemoglobin is bound to 3 molecules of oxygen. 3 out of 4 of the heme groups have oxygen on all your hemoglobin molecules. You could do it with 25% as well. So when we say 25% saturation, then we're saying that on average, every hemoglobin molecule has 1 out of 4 oxygens bound to the available sites. One thing that's interesting is how hemoglobin carries carbon monoxide as well. So carbon monoxide binds to the same site as oxygen, and it actually competes with oxygen for those sites. So if both are present in the atmosphere, you breathe it in, carbon monoxide will bind with stronger affinity and more quickly to the hemoglobin than oxygen will. It is said that it binds to hemoglobin 250 times stronger than oxygen does. And it works by displacing, knocking off any oxygen molecules that are bound. It's terrible. One thing that's interesting about carbon monoxide is that if you are exposed to carbon monoxide, which happens with smoking, it could happen due to some sort of pollution, of course, but cigarette smoking does release carbon monoxide into your lungs. It's not good. So what happens then, after you've been exposed, let's say you are having difficulty breathing and you go into the doctor and they take your hemoglobin saturation or oxygen saturation, often called pulse ox. Okay, what pulse ox means is they use this pulse oximeter, a device, which measures how much oxygen saturation your blood has. And it does so by passing a light of a certain wavelength through the skin, past all the tissues, and then to a detector that looks at any changes in the density of that light. And most of the time, the pulse oximeters have 2 different wavelengths of light that they send through. And they look at, anyway, one wavelength absorbs, detects light that has passed through blood that contains deoxyhemoglobin. And another wavelength passes through the blood and it detects oxygenated hemoglobin or oxyhemoglobin. What this means is that if you're only using a 2-wavelength pulse oximeter, the amount of oxyhemoglobin is overestimated because CO, carbon monoxide, absorbs light at the same wavelength as this oxyhemoglobin. So it could be a high pulse ox reading even if you've been exposed to carbon monoxide and that has displaced your oxygen. There are now 4 wavelength detectors that can differentiate between oxygen saturation and carbon monoxide saturation, so that is becoming more widespread. Okay, let's talk about how hemoglobin loads up the oxygen and then unloads it, drops it off. It turns out there's a positive feedback process involved with both. And for loading, what that means is that when one oxygen binds, the hemoglobin changes its shape to increase its affinity for more oxygens. That will happen every time it binds. So it's bound to 1, it has more affinity for the second. It's bound to 2, it has more affinity for the third. It binds to 3, and it has more affinity for the 4th. But it works in reverse as well, which largely deals with the partial pressure of oxygen available as well. But as one oxygen is released, hemoglobin changes its shape and it tends to lose the next one, and then it loses the next one, it loses the next one, which really is due primarily due to how much oxygen is even available. So we'll really focus on that more. But it's called cooperative binding, this concept of positive feedback where once one oxygen binds, another one wants to, etc. It's called cooperative binding. And this graph that you're looking at is called an oxygen hemoglobin dissociation curve, meaning they break apart. But you could just as easily call it an oxygen hemoglobin association curve, and they bind together. And I'm going to introduce it that way, and then we can go in reverse later when we need to. So if you just look at the red line indicates the percent saturation of hemoglobin at different PO2 values. So on the bottom, we have different partial pressures of oxygen from 0 to 100. And in the lung, you know that the partial pressure in the alveoli is about 100. In the systemic tissues, the interstitial fluid's about 40. And then on the left, you have the percent saturation of hemoglobin molecules from 0 to 100%, where 25% means that there's just one oxygen bound. And then at 50% there's 2, and at 75% there's 3, and at 100% there's 4. You get the idea. And I guess one of the most important features is the concept that this is an S-shaped curve. And this steepness right here between the arrows I drew shows you that small changes in PO2 values from 20 to 40 causes big changes in saturation. So if you're at PO2 of 20, there's one oxygen bound. But because hemoglobin is changing its shape to facilitate more oxygen binding, and because there's just a small increase, 10 millimeters of mercury, to get up here of PO2, that takes you all the way to 50%. So you went from 25% saturation to 50% saturation just by changing the PO2 by 10. And if you change it by 10 again, you get all the way up to about 75%. It takes a little longer to get to 100%, and we'll look at why that is. So I think it makes sense. There's two main points that this curve should point out to you. The first one is obvious, and that as PO2 increases, so oxygen concentration goes up, we'll just say, then hemoglobin becomes more saturated. But also notice that this is not a straight diagonal line from 0 in the lower left corner to 100 in the upper right. So that brings up a second point that's important. And that is, as hemoglobin binds to one oxygen, its ability to bind to another oxygen increases. And that's why the curve is more vertical here. It's actually a curve instead of a straight line. So at very low PO2 values, when hemoglobin binds to one oxygen, its affinity for oxygen increases by twice the amount and is able to bind more. Likewise, at high PO2, when 2 oxygens are bound, we'll say, or 3, it still has more affinity for more oxygen. It's very cool. So another consequence of this cooperative binding has to do with dissociation instead of binding to oxygen. We're going to talk about releasing oxygen now. So note that, I'm going to do this number 1 here. Note that because the PO2 is at 40 millimeters of mercury in the systemic tissues, you can see that hemoglobin only loses 22 to 25% of its oxygen. That means that 75 to 78% of the oxygen that hemoglobin is carrying is not released, at least under resting conditions. It stays in the blood. And this excess of hemoglobin, or oxyhemoglobin in the blood, I should say excess of oxygen in the blood, is called the venous reserve. That can be called upon if there's some sort of traumatic need. So that's really cool because it tends to want to bind more, right? Okay, also at high PO2, a very large change in PO2 from 100 to 80 causes a very small change in hemoglobin saturation. Only like 2% change. That is the opposite of what we saw at lower PO2s. At low PO2s, you have that much of a change, you're going to get a big difference in saturation, like a 40% difference. But when you're up at high PO2s, you get a very small change in saturation, which is perfect because where you want oxygen to be released is down here when the blood is exposed to low PO2s. It's very cool. So this is important in our systemic tissues, like think muscle, for example, metabolically active tissues where oxygen has been consumed, right? And so in metabolically active tissues, you might have as low as a PO2 of 20. And in resting, it's normally P40. But in both cases, it's going to facilitate oxygen binding to the hemoglobin. There's other things besides PO2 that influence how well hemoglobin binds to oxygen. So we're going to look at those now. We've already looked at PO2. I do want to mention just one other thing about it, and that is you can get quicker oxygen loading or unloading depending on the steepness of the pressure gradient. So we're going to look at this second point, the second factor. And I'll show you two examples. Case A and case B. In case A, we have a very steep pressure gradient. In the lung, we're in the lung in both cases, but in the lung, the alveoli have a partial pressure of oxygen equaling 104. Whereas the pulmonary artery and capillaries have a PO2 of 40 because this blood has come back from the systemic tissues. It already dropped off its oxygen. That's a big difference, right? So what is it, 64? Obviously, oxygen is going to diffuse into the blood. It's a very steep gradient, and it's going to diffuse quickly. If you look at the pressure gradient shown in case B over here, now there's less oxygen in the lung. Maybe you're at high altitude or something. And so if you have a PO2 of 80 in the lung and a PO2 of 40 in the same vessel, pulmonary capillary, you're still going to get oxygen diffusion, which is wonderful, but it will be not as rapid and not as much, right? So a higher or a steeper gradient will increase the amount of oxygen that diffuses into the blood, right? And furthermore, the higher the blood PO2, that will help enhance further oxygen hemoglobin binding. So when you have this lower, or I should say more gentle pressure gradient, it's true that it won't diffuse as quickly, and maybe not as much, but the fact that any diffuses at all helps facilitate more diffusing. It's wonderful. Okay, we're going to go back and look at these other three factors now. How blood pH and partial pressures of carbon dioxide affect oxygen saturation. So it turns out that actually concentrations of carbon dioxide play a role in how much oxygen hemoglobin can bind to as well. So first let's do pH. So when pH drops, that's an acidic condition, and it does weaken the bond between hemoglobin and oxygen. This is referred to as the Bohr effect. I don't know if I care that you remember that, but hemoglobin is normally, when it's not bound to a gas, bound to hydrogen. When oxygen is around, it binds to oxygen and you get oxyhemoglobin. But that displaces this hydrogen. So there was a hydrogen on hemoglobin and now it's free. This reaction is important because if you think of the fact that there are other sources of hydrogen in your body, we produce acid all the time. So we produce hydrogen ions, and if there's an increase in hydrogen ions, which means a low pH, this reaction is going to proceed to the left. What that means is at low pHs, hemoglobin is going to release its oxygen. The reason this is important is that in the interstitial fluids and in the venous end of the capillaries, that's where pH is the lowest. It's perfect though, because that will unload the oxygen to these tissues and to the blood, whatever. So at high pH, the reaction is going to proceed to the right, of course, because the hydrogen ion concentration is lower. Now let's look at how carbon dioxide affects pH and hemoglobin saturation. When you have increased carbon dioxide around, it does lower hemoglobin's affinity somewhat for oxygen, slightly by changing its three-dimensional structure. But more importantly, because carbon dioxide affects pH. And that's because carbon dioxide, the gas, combines with a liquid, water or water vapor, and that forms an acid called carbonic acid. And carbonic acid dissociates somewhat spontaneously. I don't know if it's a super strong acid, but somewhat spontaneously, and produces bicarbonate ions and hydrogen ions. So recall that in the systemic tissues, you're producing hydrogen ions and you're producing CO2. You're doing both. So pH is the lowest in systemic tissues for both of those reasons. So if we look at a hemoglobin saturation curve, we can see the effects of pH and carbon dioxide on hemoglobin saturation. Let's look at pH, and you see that at high pHs, there's quicker oxygen binding. There's not as much hydrogen around, and so you get a steeper curve. You reach a higher percentage at lower PO2s. It's not a big difference, but it's some difference. And at low pHs, 7.2, normal 7.4 by the way, 7.35 to 7.45. Actually, you had to know that, right? At low pHs, you get slower binding of O2, slower saturation, but what that means in reverse is that you're actually getting quicker oxygen release. It's very cool. Okay, let's look at, let's look at temperature now. So higher temperature basically results in decreased saturation. But what that means is that oxygen's released from the hemoglobin. And we tend to have higher temperature, not a lot, but somewhat higher temperature in the systemic capillaries. And so that enhances the unloading of oxygen. Very cool. Okay, this is probably a good point for you to pause if it's getting long for you, because the next topic we're going to go to is carbon dioxide transport, if you want to gather your thoughts. Okay, carbon dioxide transport in the blood occurs, as we've talked about already, in 3 different ways. We're going to look at how it's transported by hemoglobin a little bit, and how it's transported as a bicarbonate ion in the plasma. So I'm just going to ask you to recall this introductory slide that I showed you at the beginning. It was a little bit different, but basically it shows how carbon dioxide is transported in 3 different forms or locations in the blood. 10% of it is dissolved just as CO2 in the plasma. Higher amount than oxygen in the plasma, though. And 20% of it is as carbaminohemoglobin. That means bound to hemoglobin. What we're most interested in is the majority, 70% of it, is in the plasma as this bicarbonate ion. So let's review one more time. You're going to get sick of seeing this. How bicarbonate is formed. I think you get the idea that this is an important reaction, right? Carbon dioxide combines with water to form carbonic acid. Carbonic acid is H2CO3. That dissociates into hydrogen ions, and there's the bicarbonate, HCO3. Now this reaction does occur in plasma. So any of the carbon dioxide that enters the plasma, the 10% that stays in there, some of it will get converted, but not a lot. And it is measured in the 70% anyway. The majority of bicarbonate is formed in erythrocytes. And that's because the erythrocytes have an enzyme called carbonic anhydrase that lowers the activation energy needed for this reaction to take place. So there's an enzyme that facilitates this chemical reaction. So what you see is carbon dioxide entering the bloodstream. It first enters the plasma, then it goes into the red blood cell where it combines with water to form carbonic acid, which spontaneously dissociates into these 2 ions, bicarbonate and hydrogen ions. But that does not explain how so much bicarbonate gets into the plasma, because 70% of all carbon dioxide that enters the blood is going to be in the form of bicarbonate out here in the plasma. So we're going to look at that next. And this shows you how bicarbonate moves from red blood cells into the plasma. So let's start with the systemic tissues, because that's where we have a whole bunch of CO2 that has been formed. It diffuses into the blood, and in the red blood cell we get the formation of this bicarbonate ion. And it turns out that there is a membrane protein in the red blood cell's membrane called a chloride bicarbonate exchanger. It doesn't matter. You can think of it as a transporter or a channel. And it, by facilitated diffusion, moves bicarbonate to the plasma in exchange for chloride coming into the cell. They're both anions. They're both negatively charged, but it's the bicarbonate that moves to the plasma and the chloride ion that comes into the red blood cell. And I bring up this movement of chloride ions into red blood cells because you'll probably encounter it in your professional health program. It's called a chloride ion shift, and it illustrates the importance of electrolytes that contain chloride, like sodium chloride. Sodium bicarbonate is also important to the sodium chloride content, but it just shows you that chloride is important, and when we study the digestive system, it'll come up again. Okay. So now this bicarbonate is in the plasma. It's traveling in the venous system. It got picked up at the systemic tissues, right, in the capillaries. Let's look at how this hydrogen that was produced also plays a role. This hydrogen that came from the carbonic acid, how does that play a role? Considering where we are in the blood. So remember, in the systemic tissues, this is where CO2 is really high. And presumably oxygen is low because the oxygen has been used to make ATP. So it turns out that after carbon dioxide comes into the red blood cell and carbonic anhydrase makes this carbonic acid, we get bicarbonate that's going to be carried in the plasma, going all the way back up to the heart and then to the lungs. The hydrogen plays another important role. And presumably, since this blood came to the capillary bed full of hemoglobin bound to oxygen, it's very important that at the same time it's taking up oxygen, the red blood cell I mean, taking up oxygen, at the same time that hemoglobin is taking up the carbon dioxide, or the red blood cells taking up carbon dioxide, the hemoglobin needs to release oxygen. I'm going to say that one more time. I'm very sorry to make you listen to me again. But at the same time that this red blood cell is taking up carbon dioxide, it needs to release oxygen. And that oxygen is bound to hemoglobin, the majority, 98.5% of oxygen in the hemoglobin. So this hydrogen that's been produced will displace the oxygen, and now the oxygen is free. And there's a pressure gradient. So when there's high O2 in the blood but low O2 in the tissues, it's going to leave the bloodstream. Perfect. Okay, so what we've got is bicarbonate traveling in the bloodstream back to the heart and getting pumped to the lungs by the pulmonary artery. We need to see how that bicarbonate translates to us exhaling or eliminating the carbon dioxide. So, how does this bicarbonate produce CO2 in the air we exhale? How does that translate? So now we're in the lungs, and I have my little alveolar sacs and alveoli over here. And we've got the bicarbonate, and we need to get rid of that. That needs to get out of the bloodstream. And so we see that actually the reaction just happens in the reverse. You actually get a reversal of the chloride ion shift too. So chloride ions will leave the red blood cell, go out to the plasma, and the bicarbonate will be taken up by the red blood cell, where it combines with hydrogen. Because there's an enzyme in the red blood cell called carbonic anhydrase, this happens quite readily and gets converted back into carbon dioxide, and water. The water stays in the blood, but the carbon dioxide will diffuse out of the red blood cell into the plasma and then out of the plasma, out of the vessel entirely, because there's very low CO2 in the alveoli, but we've just made a whole bunch of CO2 in the lung. So the chemical reaction that we just studied in the systemic tissues in one direction happens in the opposite direction in the lungs. It's always happening in the red blood cells. It's just that the red blood cells perform the opposite or reverse reaction in those two different locations. And it's driven partly by the concentrations of hydrogen ions, CO2, and oxygen. And we're going to talk about that next. We're going to put it all together. And so again, I would suggest that if this is getting long for you, this is a very good time to pause, because the next couple of slides will be a review, not just of what we talked about here, but also what we talked about with partial pressures in the first lecture on gas exchange in the lungs and the systemic tissues. Kind of pulling it all together. It's not that much more, but you might want to take a break. Okay, this is a summary of gas loading and unloading that happens at the systemic tissues. We're going to start at the systemic tissues because I find that it makes the most sense for students to start here. Now, I've provided this slide for you, but I've also given you a slide that has all these items blank. And my suggestion is to not fill them in right now. And we'll come back to what I think you should do with this blank slide in a minute. So what we're going to do is cover what happens to oxygen and carbon dioxide as they move between the interstitial fluid of the systemic tissues and the bloodstream. And I think besides knowing you're at the systemic tissues, the first thing that you need to do is think about what the concentration of carbon dioxide and oxygen is in the tissues as well as in the blood. You don't have to be exact, but you should know where it's high and where it's low. And so we know in the tissues that PCO2 is high because they've been making ATP, they've been producing CO2 because they're breaking down all these nutrients, and O2 is low. But in the blood, the opposite is true, because the blood that has arrived to the systemic tissues before it takes off again is carrying oxygen on hemoglobin, and it doesn't have a lot of CO2 in it because that was just exhaled. So we're going to start with this first point, and that is that in the systemic tissues, CO2 is high compared to the blood. And so CO2 is going to diffuse into the plasma. Some of it will stay in the plasma. Some of it will get converted in the plasma very slowly, but to carbonic acid. Some of it will move into red blood cells and bind to hemoglobin, a very low percentage. Most of it, and this is what we care about the most of the CO2 will move into the red blood cell, combine with water, and because of carbonic anhydrase, the enzyme, it'll get converted into carbonic acid, and it will dissociate. Carbonic acid will dissociate. So now we have two anions. One anion and one cation. Let's talk about the anion, the bicarbonate. The bicarbonate is shifted to the plasma in exchange for chloride. So chloride comes in. We're not going to do anything with the chloride. Now we have this free hydrogen here. that lowers the pH. So it was pH of 7.3, now it's a pH of 7.25 or whatever. It actually doesn't get that low. But anyway, the free hydrogen ion can displace the oxygen that's on hemoglobin. Perfect. Now we've got free oxygen in number 8 here. The hemoglobin releases the oxygen. And now, because PO2 is low in the tissues, there's this gradient to cause the oxygen to diffuse into the tissues. So we kind of came full circle. The CO2, by way of making this hydrogen, has actually helped the release of oxygen and get the oxygen to the tissues. It's wonderful. So my recommendation on studying this and having this make sense to you, it's the only way you can do it, in my opinion, is to go through it step by step. And my recommendation is to print the slide out or take a picture of it or whatever, and not write in those blanks. You can use a separate sheet of paper to write down what happens first, second, third, and fourth, or draw your own diagram. And always start with, high or low CO2 and O2. So in the tissues, CO2 is high. And in the tissues, PO2 is low. But in the blood, CO2 is low and oxygen is high. That's what happens at systemic tissues. And then just go through the numbers that I've provided for you. And we're going to do it in the lungs. We're going to look at what happens in the lungs, a summary of the last 2 lectures, I think. So this is gas loading, unloading, and transport in blood that reaches the lungs. So blood that's reaching the lungs has low oxygen. It just dropped it off. And it has very high CO2. The CO2, of course, is in the form of bicarbonate. But the blood is encountering a new environment, sort of, and that is, or exposure to, the alveoli and new pressure gradient. And in the alveoli, oxygen is super high, and we're going to see that's what drives everything. In the systemic tissues, it was high CO2 that drove the mechanisms. Now we're in the lung, and it's high O2 that drives the mechanism. So when O2 is high in the lung and low in the blood, it diffuses into the plasma. We know that some of it, 1.5%, stays dissolved in the plasma. But the majority of it will bind to hemoglobin. And it was deoxyhemoglobin to begin with, HHB. And now it's going to become oxyhemoglobin. That displaced a hydrogen ion again. This silly hydrogen ion keeps popping up, right? Well, the free hydrogen ion is going to become important because it's going to be needed to convert the bicarbonate into carbon dioxide. But I don't want to get ahead of myself yet. Let's go up to number 4 because you're going to need this for number 5 to happen. And that is that now when we're in the lung, the chloride ion shift happens in reverse and bicarbonate comes in to the red blood cell. Now I think you can see why the hydrogen is important. The hydrogen ion will bind to that bicarbonate, and because we have carbonic acid or carbonic anhydrase around, it converts the carbonic acid to carbon dioxide and water. Now we finally have our carbon dioxide, which before existed in the plasma as bicarbonate. So the carbon dioxide is now high in the blood and low in the alveoli, and so it will diffuse into the alveolus. Now, due to oxygen uptake, because PO2 is so high, that will help hemoglobin dissociate from any carbon dioxide it was bound to. In addition, any carbon dioxide that was dissolved in the plasma, it will actually diffuse into the alveolus too. But that's just because PCO2 is low in the alveolus. Again, we have a slide with blanks for what happens when blood reaches the lung. And again, I suggest that you not fill these in, use a separate sheet of paper, and draw diagrams as you need to make this whole gas transport, loading and unloading, make sense to you. Thank you very much. That's the end of this lecture.